Good day to everyone joining us, and welcome to today's X-Talks webinar. Today's talk is entitled Enabling Clinical Development of Poorly Soluble Molecules Through Formulation Solutions. My name is Ryan Muse, and I'll be your X-Talks host for today. Today's webinar will run for approximately 60 minutes, and this presentation includes a Q&A session with our speaker. Now, the webinar is designed to be interactive, and webinars work best when you're involved. So please feel free to submit your questions and comments for our speakers throughout the presentation, and we'll try to attend to your questions during the Q&A session. This chat box is located in the control panel, which is on the right-hand side of your screen. And if you require any assistance, please contact me at any time by sending a message using this same chat panel. At this time, know that all participants are in listen-only mode, and please note that the event will be recorded and made available for streaming on xtalks.com. At this point, I'd like to thank Aragen, who developed the content for this presentation. Aragen Life Sciences, formerly GVK Bio, is a leading R&D and manufacturing partner, CRO, CDMO, for life science firms worldwide. With a team of over 3,100 professionals and through a network of sites around the world, they offer a seamless, integrated platform to advance customer programs from discovery through commercialization for both small molecules and biologics. Established in 2001, Aragen now serves over 450 customers worldwide across multiple modalities and therapeutic areas. Now, I would like to introduce our speaker for today's event. Sreehari Putchakayala has over 18 years of experience in pharmaceutical development, with 12 years in leadership roles covering early phase drug discovery support, product development in drug discovery R&D, generics formulation development, and clinical supplies management. Sreehari previously worked with Novartis Pharmaceuticals, Ranbaxy Research Laboratories, and Dr. Reddy's Laboratories, Hyderabad. And so now, without further ado, I'd like to hand the mic over to our speaker. Sreehari, you may begin when you're ready.
Good morning, good afternoon, everyone. This is Sreehari Babu, connecting from Hyderabad. Thank you all for joining this webinar, and thanks, Ryan, for the kind introduction. So today I'm going to cover a broad overview of what formulation solutions can do for a poorly soluble compounds in the drug discovery and development. So basically, how do we enable clinical development of poorly soluble molecules to appropriate selection of formulation solutions? So next slide, please. So broadly, the agenda includes a brief introduction on why appropriate selection of formulation solution is important at early stage of the discovery process, and what are the types of formulation techniques available for developing molecules to clinical phase, and in a little bit more detailed introduction of each of these techniques in the next phase. Next slide. So this is the broad drug discovery and development phases from lead optimization to the phase 3, and the formulation plays critical role at every phase, working together with chemistry, DMPK, and late phase chemical development and formulation development teams. Next slide. So this is a preclinical formulation R&D and clinical formulation R&D at tox phase, and followed by clinical manufacturing and GMP analytical testing. So today's overview is mainly focused on preclinical formulation R&D, where we intervene the formulation with the right formulation approach at early stage of the compound discovery and early stage of development. Next slide. So what are the typical issues observed during NCE development, which causes the dropping of molecules or making it delayed to the development phase or delayed to the clinical phase? So usually it's poor aqueous solubility and poor bioavailability and poor physical and chemical stability, and low permeability, first pass metabolism, and short half-life and duration of action. These are, I'm only covering the issues we faced in terms of pharmacokinetics and all. There may be many other issues, target related and efficacy related issues. But the right formulation approach can help in the first three areas: improve aqueous solubility and improve the bioavailability, and also stabilize compounds in terms of physical and chemical stability. Next slide. So this is to just give you an overview. So how do drug delivery systems influence the bioavailability? There are some compounds, several of BCS class 2 or 4 compounds are they're really infallible, just like Victor. So with solubility less than 1 microgram per ml, to be able to get a reasonable dose of 100 milligram into the systemic circulation, we require huge amounts of liquid to dissolve the drug, which is impossible to in vivo environment. So either we have to stop the compound here for such compounds, or we have to identify a suitable drug delivery system which can enhance the solubility to a level where it can have a decent systemic bioavailability. Next slide. So this is taken from next two, three slides are taken from the literature. So this is a broad understanding of what is the drug discovery, development, attrition. So we start discovery with so many compounds, and then eventually leads to only one FDA approved drug out of so many compounds discovered and tested. So how do we improve this rate of success from one compound to just having two FDA approved compounds for the same amount of effort can have a dramatic impact on the drug discovery, and then so this is the same giving same indication of what we are discussing before. So these are the typical reasons for the drug profile. So lack of efficacy is one of the predominant which we cannot address through formulation, but the highest reasons are usually poor PK and ADME properties of the compound, which we can address through formulation solution. So you can say that nearly 40% of the compounds can be improved on their profile through a right formulation approach. Next slide. So this is BCS classification you might have all familiar with. So the right class 1 compounds are easy to develop, does not require much effort, and class 3 compounds also formulation cannot improve much on this. So it's usually if the permeability is too low, it should be the compounds need to be changed to a better permeable compound. So formulation solution can improve the class 2 and 4 compounds, which are unfortunately majority of the current development pipeline and also in the marketed compounds. So this is where the focus is. Class 2 compounds have best chance to develop through formulation solution, and class 4 compounds, to some extent, we can improve class 4 compounds also, depending on the permeability, level of permeability. Next slide. So this is a graphical representation of what happens in a drug solution. So drug is so it is in a dosage form and dispersed into a granules and then into a particles, which and then dissolving in the dissolution medium and goes through the GI membrane, and then through the liver. It may possibility of metabolism, and then the eventual what is available in systemic circulation is the bioavailability. And next step. So the first point is so what we can influence with formulation solutions is the solubility, dissolution, And what we cannot influence is permeability and efflux of the compound, which are nature of the compound, and metabolism and elimination. So these are the things which we cannot control. And the first part is what is happening before compound is absorbed through GI membrane is something we can control through formulation approaches. So it's very important at early stage to understand the molecule to see what we can do to the compound instead of jumping into directly formulation development without understanding compound nature. Next slide. So what does drug delivery systems do? Drug delivery systems can enhance the absorption, control the drug release, reduce variability, and target the systemic delivery to a certain target area, and introduce mechanical devices as required, and increase the patient convenience and overall better therapies to the patient through right approaches. So there are some formulations we just develop only to reduce the variability, not to enhance specifically solubility or absorption. It's just the formulation gives much more uniform across the patient. It reduces patient variability, giving a better dosage regimen to the patient. Next slide. So this is a broad overview of what we do. We get the drug substance with certain properties. So we need to first understand the properties of the compound, what is limiting its successful development, and devise the appropriate formulation strategy to get through the preclinical phase, and then utilize the knowledge we generated in the preclinical stage and adopt the right formulation approach for the clinical phase, depending on the route of the delivery. And solubility challenges remain for every route of the delivery, including injectable. But anyway, this webinar is mainly focused on how to enhance the oral bioavailability of the compound. But solubility play an important role in any route of the delivery. So overall drug discovery and development is a cross-functional effort. To develop an appropriate formulation and select a right approach for the clinical and pre-tox studies. We have to work with several teams with the discovery chemistry for compound selection. Sometimes the first option to improve the compound properties is to modify the structure, if it is possible to allow, instead of directly working on the formulation approaches. If we can improve by small minor structural changes the physicochemical properties of the compound, that's the best approach to develop a compound. And we have to work with chemistry teams to understand possibilities of changing structure without compromising on the efficacy, and followed by working with DMPK teams to profile the compounds in different formulations and the different species, and select appropriate formulation strategy for the compound. And then later with the tox experts to select a safe formulation and which shows sufficient exposure multiples in the tox study, and transfer all this knowledge to the later phase, to the chemical development and formulation analytical development team, to translate into a perfect clinical formulation strategy to enable the final development of the compound, successfully finish clinical phases of the compound, fully soluble molecule. Next slide. So that's a broad introduction to try to give you overall feel of why it is so important to engage formulation teams to select the right formulation approach at the earliest of the compound development, so that to avoid surprises and delays in the drug development, or compounds getting killed or at a late phase, which will cost the companies much more than a bad compound killed at early stage. So here I will spend my next few slides more on the technical part, where the formulation solutions are required during the drug discovery and development phase. So in the early stage, the first three parts are discovery phase, and then clinical is the most development phase. In the efficacy stage, we develop a fit for purpose formulation. So ideally, solution formulation is preferred to support efficacy studies, which gives the best possible exposure for the compound, and also just to understand the compound is efficacious enough or not for further development. So it may not be safe formulation, may have a higher level of excipients, but it will be the fastest way to have enough exposure in systemic circulation and get efficacy readout to see whether compound is has required properties to pursue other activities. So once compound is proven to efficacious, then the next optimization is the PK profiling in different species, in mice, rat, and the dog or cyno, to understand the physical form of the compound and evaluate solution versus suspension to understand solubility limited absorption or dissolution limited absorption. And this is the PK profiling is the basis to select the right tox formulation approach or strategy to have a more safer formulation, and eventually going to toxicity formulation, which may also involve enabling formulations for very poorly soluble compounds and achieve required exposure multiples, and safety of vehicles is also critical in the toxicity study. And in the clinical phase, many of the compounds will go with the tox and clinic same formulation strategy, but depending on the dose regimen, and which may change dramatically from tox to clinic. So we may sometimes go back to conventional formulation in the clinic, depending on the clinical doses expected for the compound. Next slide. So this is the initial phase when we get a compound first from chemistry team to understand the compound. What we do is a compound personality assessment. Besides this data, there are a lot of physicochemical properties like pK, ionization constant, log P. Other data is generated. I'm just covering what we generate in the formulation perspective. So we understand the solid state of the molecule in terms of crystallinity, melting point, is it high, low, or medium, is it hygroscopicity and crystal habits of the compound, and followed by solubility, pH solubility, and in biorelevant media. So the usual goal of solubility is in a biorelevant media is above 100 microgram per ml. This is in terms of clinical point of view. But for toxic studies, depending on the dose, we may require a much higher solubility than this one. And stability of the current form as is in the bulk powder state and also in the solution state, to understand, to be able to predict shelf life of the molecule and also gastrointestinal stability of the compound. There are many compounds which are highly unstable during the course of absorption, the GIT. So that's also understood in the early phase itself to avoid efforts which are going in a different direction while the compound is unstable in the medium. So this gives us overall understanding of compound properties, whether we should focus on improving solid state properties or solubility or stability of a given compound. Next slide. So the objectives of the right formulation selection for a poorly soluble compound is mainly to improve solubility and bioavailability of class 2 and 4 compounds. By the time we start working on these compounds are already classified as class 2 or 4, and those compounds are only suitable for these approaches. And to enable right formulation selection for toxicity studies to achieve required exposure multiples, which are 5 times or over 10 times of the efficacious AUC levels. And finally, to assess the developability of NCE candidates for clinical phase. So it's better to know as early as possible if this compound can be developed as a clinical to the clinical phase with appropriate formulation approach. Next slide. So the typical solubility enhancing approaches listed here, or with increasing to the right side, is increasing complexity and also increase the rate of success. The first one is a physical form approaches, where we can form a salt or co-crystal for compounds depending on their nature of ionizable or non-ionizable compounds. And these are well-known literature and well-known our own experience shows that they can improve significantly pH-dependent solubility and also dissolution of the certain molecules to some extent. Followed by conventional formulations like suspension, where we just use particle size reduction as an approach to, and also include small amount of surfactants, which can help also achieve moderate improvement in the bioavailability. And followed by solution, which is the simplest formulation approach, but can give dramatic improvement in the bioavailability for several compounds. Basically, the idea is this straightforward formulation. We just need to find the right excipients to dissolve the compound and form a non-precipitating solution formulation, which can has a possibility of giving the best bioavailability, and also the simplest formulation to achieve complete preclinical phase of development. And towards the right are more complex formulations in terms of manufacturing for tox and also for clinic, but have a better rate of success for very poorly soluble compounds. So microemulsion lipid-based formulation suitable for highly lipophilic compounds. And the nanosuspension, where particle size is reduced to the nano size to achieve the highest surface area and the dissolution. And amorphous solid dispersion, which is converting a crystalline drug to amorphous drug and stabilizing with a polymer. So these are the broadly techniques available, which can be taken commercialized also if required, but can be supported for tox, clinic, and also for commercial. Next slide. So these physical form alteration approaches. Sorry, previous slide. So salts are basically the ionizable compounds, whether it is acidic or basic compounds, are reacted with a suitable counterant in presence of solvent and the crystallization process, and so we try to find the optimal salt form which can have better solubility or dissolution rate. And cocrystal are for non-ionizable neutral compounds, can also form cocrystals. And they are known. The chances are usually lower than the salts, but can if we can form a appropriate cocrystal, they can still have a better exposure and achieve moderate improvement in bioavailability. Next slide. So these are the usual solid forms of the API. The single crystalline compound, a single component, and clean form. But when we have issues of solubility or stability issues, then we try to form it into or maybe a multiple polymorphs. We try to form a salt or cocrystal of the compound, either to improve their solid state properties, and we can use the same approach for improving solubility as well and dissolution rate. So both are well-known techniques, very simple to make. It doesn't require much formulation efforts for tox or clinical supply. Just a small moderate changes in the drug itself. Next slide. So this is a typical salt co-crystal screening. It's designed based on right selection of counterant or coformer and the appropriate solvent depending on the solubility of the compound itself. So this is a flexible screen model. It can be done as many counterants and solvents as possible. depending on the compound, and can be done in a high throughput model with a very less amount of material, less than a gram material can be utilized for high throughput screening. And the hits found from this screening are usually characterized fully and selected. The best best salt or Cocrystal is selected for the PK studies if we see the in vitro solubility or improvement. Next slide. So this is the typical in vitro evaluation of salts or Cocrystal. So you can see that this is the first column is the free form solubility, which is has a decent solubility at low pH, but poor solubility at higher pH. So when we form a salt with and they can be profiled in the same media, slightly improved solubility because of pH modification in biorelevant media and higher pH. And the same is again confirmed with intrinsic dissolution rate. Also, that shows that the wettability and dissolution rate is much higher than the free form. So this is in vitro evaluation, followed by we confirm the same data again at the suspension state study in animals. The next slide. So in vivo evaluation, so the rate of increase can be different from each compound, but it can be for some compounds we can achieve 2 to 3 x of the exposure, depending on the molecule. So if that is not enough, then we usually go to novel formulation approaches. But false Cocrystals would be the ideal first approach to improve the in vivo exposure bioavailability for a compound, class 2 and 4 compound. Slide. The advantages are relatively easy approach to resolve solubility dissolution challenges. These require no additional formulation efforts for tox or clinical manufacturing. Provides IP protection. All salts, polymorphs, Cocrystals found can be patented in addition to the molecule patent. So limitations are moderate solubility, bioavailability improvement. It's not suitable approach for all compounds, and merely forming a salt Cocrystal does not guarantee solubility improvement. But that's the first step of the entire process of evaluating in vitro and in vivo to understand. But success rate is a bit lower, but those salt formation, Cocrystal formation can be achieved with high success rate, but not all salts can achieve tox exposure multiples required for many compounds. So that's physical form alteration of approaches. The next techniques are all mostly the formulation approaches where the compound is completely transformed through formulation techniques. Either they are dissolved or reduced particle size to very low or converted to amorphous. Next slide. So this is the first of the simple formulation approach, conventional formulation, the suspension formulation with only the particle size change to a micron size, reducing to 5 to 10 microns. That's for several compounds which are borderline solubility. That particle size reduction itself can work in presence of small amount of surfactant, can help to improve the exposure significantly. It's relatively easy to prepare the formulation, can support same approach up to the clinic as well, where we can use a micronized drug for the tablet or capsule formulation, but only suitable for compounds which are borderline and have a decent enough solubility on their own. Next slide. So this is a relatively easy approach to improve dissolution or wettability. Can be translated into regular solid oral formulations, require no additional formulation effort for tox or clinical manufacturing. So the limitations are It's not suitable for solubility limited compounds. So only can improve a little bit dissolution rate. And these are also limited by the physical form changes. But a compound's ability to withstand the milling or the sonication activity. So those compounds are, many compounds can convert to amorphous form during the process. Next slide. So this is the solution formulation, most common, commonly used and first preference formulation, relatively easy to develop. It involves a compound is completely dissolved in a suitable excipient, which involves surfactants, co-solvents, buffers, and polymers, or complexing agents like cyclodextrin. So it all depends on compound's ability to dissolve in certain vehicles like these. If the compound have enough solubility, we can develop a non-precipitating solution formulation. If it doesn't have a solubility, we can't do much about it. It's more straightforward, go kind of formulation approach. If it works out, it is a much easier formulation to go all the way to top, and maybe even for phase 1 as a liquid formulation. To proof of concept in man can be achieved very fast for compounds in this approach. So once we develop a solution formulation, we can also test solution versus suspension to understand the molecule solubility limited absorption. Usually compounds with solubility limited absorption will give much higher exposure in solution state, assuming that there is no metabolism and permeability issues are involved. Even though we evaluate in the in vitro so far on the solubility limitation, but this study gives the best outcome in terms of conclusion that a compound has purely solubility limited absorption, or any permeability or metabolism is also involved in the lower bioavailability of a given compound. So next slide. The advantages are it's a relatively easy approach to improve bioavailability. So it does require certain expertise to develop a formulation, but compared to other approaches, this is much easier to prepare, and requires no additional formulation effort for tox or clinical. It can be prepared at the dosing site. Suppose for tox studies, it can be prepared same way it's prepared as a suspension formulation. It does not require specific instrumentation or equipment. And limitations, it's not suitable for all compounds. It depends on the compound solubility in the acceptable vehicles used for tox studies. And these formulation approach carries relatively high organic load. It involves a lot of surfactants and co-solvents, which can have its own tox issues or adverse events in the tox study. And possible stability challenges in the solution state for certain compounds. So it's not a challenge for tox study supporting, but it can be a challenge for clinical phase, where it requires a little bit longer stability for the formulation. So in the eventually it becomes challenging to deliver for clinical studies in terms of stability itself. And being a liquid formulation, but the preparation itself is much easier from preparing the formulation. So these are the two conventional approaches, suspension and solution. This compound can be developed with those approaches. That's the best outcome for a compound. But once we evaluate the physical form approaches, conventional form approaches, if none of them works out for a compound, we usually go into more complex formulations, which we call novel drug delivery systems. Some of these we may not need to do all the formulations in parallel, but they can be customized to the compound properties. For example, microemulsion is only suitable for compounds with high enough log speed. So, and solid dispersion can be also very difficult for a compound with very high melting point, poor organic solvent solubility. So some can be customized based on compound properties, but at least one or two formulations must be evaluated in practice to understand the best formulation approach to develop that particular compound. So microemulsion involves complete dissolving the compound in a liquid phase and stabilizing with a surfactant. And this, if a compound has a decent solubility in liquid vehicles, so this is the best approach, can have a guaranteed bioavailability improvement. And nanosuspension is reducing the particle size of compound to nano range to around 400 to 500 nanometers, which increases surface area and dissolution rate dramatically. And finally, the amorphous solid dispersion, one of the most widely used technique, and also works well for higher percentage of compounds in general, because theoretically many compounds can be converted to amorphous state. We just need to select right polymer and surfactant to stabilize the system and achieve the required exposure level. Next slide. So I'll go through the techniques briefly about each technique. So microemulsion is a class of self-emulsifying drug delivery system, which is formed spontaneously during the preparation, and it continues in the system after dosing. Also maintains the microemulsion state, this is almost like a solution formulation, but technically is not a solution. But the drug is in a liquid phase and with very low droplet size of much better than a milky emulsion. Somewhere it falls between micelle and regular emulsion. And this doesn't, so this is completely, so the solid form is completely dissolved. So the effect of solid form is completely eliminated in this approach. So it's only appropriate for compounds with high enough log P and which are screened in different lipid phases to select appropriate oil vehicle to achieve the microemulsion formulation at the required dose range. So the development involves screening multiple surfactants, oil phase, co-surfactants, co-solvents, and form a emulsion which is almost clear visually. It's almost clear, but it is always translucent kind of one. It's different from visually different from solution formulation with very low droplet size and physically stable upon dilution with GI relevant media. And the formulation can be supported for the tox studies and also for clinical studies, either filling into capsules or it can be supplied as a liquid in water. So the next slide, please. So in vitro evaluation is not much for this compound. It's only about developing a formulation and checking the dilution stability in the biorelevant media. And once it is established, it's non-precipitating emulsion, then we can conduct in vivo evaluation, and you can see the typical how if it is a compound which has high lipid solubility, it is the best approach, can give very high exposure level. You can see the comparison of one of the molecules where the usual tablet or a suspension formulation is the lowest, followed by solid dispersion, and microemulsion gives the highest exposure, especially the Cmax. So it gives the best exposure because of the very strong system inside containing high level of lipid and surfactant. But again, the safety of vehicles and balance. So many times we evaluate multiple formulation and we may still select the solid dispersion given that it is a solid dosage form and for clinical phase and vehicle safety, etc. Depending on the molecule, we can select the appropriate formulation approach. Next slide. So microemulsion is a relatively easy approach to improve bioavailability, requires less complex manufacturing efforts for COX or clinical manufacturing, and limitations are only suitable for highly lipophilic compounds, and it's a relatively high organic load and the possible stability challenges. So many compounds are also unstable in presence of in solution state. So it will give shorter shelf life and stability, especially for clinic. Next slide. So I have next a few slides. I will cover nanosuspension and solid dispersion. So nanosuspension is basically we take the crystalline material, bring it to a micronized particle size level, either through wet milling or other approaches, and then further we nanomill it using a high intense ball mill to achieve a particle size of below 500 nanometer. So this process involves right selection of polymer and surfactant to achieve the nanomilling, and also the maintaining the physical form as is. So the nanosuspension should be the same crystalline form as the starting material to be able to have a stable system. Next slide. So the development is typically involves appropriate polymer surfactant selection. We screen multiple formulations of over 20 different combinations and select the right formulation approach which can achieve a particle size reduction and also keep the physical stability of the formulation in terms of crystallinity and also particle size. And we perform scale-up studies and stability and PK studies to evaluate the suitability. So sometimes we also, nanosuspension tend to be very species different. It works well in larger species than rodents, what based on our experience and observation. Because of it's more like a dissolution rate improvement only. It doesn't improve solubility as much. Next slide. So in vivo evolution, so it's the same level. The level of improvement can be different from each compound, but it can improve as much as solid dispersion, but lower than solution or microemulsion. Those are where the compound is completely dissolved. Next slide. The main advantages of this technique is low organic content, and limitations are not suitable for many compounds, mainly because of physical form changes or particle size regrowth during the stability. And it's a quite difficult process to convert into a proper solid dosage form for the clinical studies, and which is automatically becomes a very complex manufacturing process to deliver clinical supply. And the last formulation technique, but is the most important and most widely used technique, is the amorphous solid dispersion. So here what the crystalline drug is loaded onto a polymeric excipient, and as you can see in the animation, so there are multiple types of solid dispersion. The crystalline solid dispersion, where the crystalline drug is suspended into a amorphous polymer, which can still improve moderate improvement, and there is followed by amorphous drug dispersed in amorphous polymer, complete amorphous system, and the last is solid solution, where the amorphous drug is completely solubilized in a polymer in a solid state situation. So it's not every compound is suitable for all, but amorphous solid dispersion is the most widely used technique out of all these. But solid solutions are also possible for some compounds at up to certain drug loads. So the complexity of developments and the increase in bioavailability improves with techniques to the right side. So our goal is always to achieve a solid solution when we're developing a amorphous solid dispersion. but we may end up developing it up to a amorphous solid dispersion in many cases. Next slide. So solid dispersion development typically involves, again, polymer surfactant screening. Unlike nanosuspension, here we use a lot more polymers. And usually we do solvent evaporation process for screening. And for scale up, depending on the compound properties, melting point, etc., we either approach a spray drying process or hot melt extrusion to scale up the formulation and supply for the tox studies or for the clinical development. So basically, the polymer selection is the most critical part, and then identifying the appropriate drug load, which is miscible with the selected polymer, is the most critical part of this. Once that is locked in, the formulation can have a reasonable stability for the duration of the shelf life expected for the amorphous formulation. Next slide. So solid dispersion requires much more in vitro evaluation studies than the previous formulation. Here we evaluate solubility and also the stability extensively in the in vitro before we go into the in vivo studies. As you can see, left side in vitro study. So this is a classical basic compound which has high solubility in low pH. But when we change the pH after 60 minutes, it's rapidly precipitates, and that's why, which is intestinal pH, where unfortunately 70-80% of the drug is absorbed. So it's rapidly precipitates, and absorption is very low absorption. So we develop a formulation and try to see the in vitro models where we see the oral solution and solid dispersion see the maintains the supersaturation and maintains in solution state. And the formulations are tested in the in vivo models to further establish this improvement and select the right formulation approach based on the in vitro model. So in vitro models may not be able to predict the amount of bioavailability we may achieve in the in vivo, but it gives the rank order of the right formulation selected for the next studies. Next slide. So solid dispersion basically have a low organic content. Of course, it has a lot of polymer, but their polymers are typically safe for tox or for clinic, and suitable for a wide range of compounds. Whether it is ionizable, basic, acidic, neutral, all kinds of compounds are suitable for this technique. And limitations are sometimes high melting compounds are difficult to prepare through this technique, and typical physical instability expected in amorphous state. And it's also a complex process to convert solid dosage form for the clinic, but for fully soluble compound, that is a balance we have to achieve, whether to develop a molecule or select a complex formulation approach. So these are all the wide formulation techniques available at the clinical stage to evaluate. Some of those techniques can be translated to clinical phase, but again, depending on the dose expected at the tox and also at clinic, formulation approaches may change from complex at tox phase to a simpler in the clinical phase. But sometimes it can be same formulation approach at both phases. Depending on the compound and molecule, we can select the right approach. So last two slides. Next slide. So basically, right formulation approach can create a lot of value to the drug. The early we intervene, the better we achieve, improved success rate. At least we can also tell clearly that this compound can be developable, or will create lots of issues in the later phase. Next slide. So it also can improve the patient compliance. Sometimes we have to give lots of tablets or capsules to a patient to achieve a required bioavailability. from a regular technique, but by utilizing the right formulation approach, we can reduce that dramatically and can improve the patient compliance in the clinical phase or in the commercial phase. So I think that's all we have for today. And I'm just showing the contact details. If you have any queries, even after the webinar, please feel free to reach out to us.
Well, thank you very much for that very insightful presentation. I would like to invite our audience to continue sending their questions or comments right now using the questions window for the Q&A portion of this webinar. Now, I've already received some questions, so we'll get ourselves started with those. Our very first question for you asks, what are the typical timelines to evaluate novel formulation approaches?
So depending on whether we would like to evaluate microemulsion nano and all together or one after another, it depends on the molecule properties, amount of compound available. It can be 4 to 5 weeks, including a PK study. If you evaluate all 3 formulations together, but usually some compounds require 2 formulation approaches evaluated, and depending on the compound availability and client's willingness to evaluate in parallel to save time for timeline sensitive compounds, it would be either in parallel or one after another. It can be 4 to 5 weeks, including a PK study, which takes about a week.
Excellent, thank you very much. The next question we have for you would like to know, when is the right time during discovery phase to intervene with formulation development?
So the right time is in efficacy study when it is the compound. The first part is the compound needs to be understood. It has a solubility challenges or not. So which need a compound personality assessment, and then followed by efficacy studies is the right time to intervene with the simple formulation approaches, and then later PK profiling and then talk.
Excellent. All right. Thank you very much. The next question we have for you would like to know which technique offers the best chance for successful formulation strategy for tox and clinic?
So again, depending on the compound solubility properties, so it can be borderline to moderate to worse solubility. So the best approach, if you know the compound is really poor solubility compound, the best is directly go into novel formulation at the earliest. So that could save a lot of time and effort in serial formulation development approaches. We can go directly to say amorphous solid dispersion in the first go itself, which can have a best chance to develop a compound.
Excellent. Thank you very much for that answer. The next question asks, is it possible to select right formulation approach based on compound properties?
Yeah, it's possible. We may not be able to single out a one formulation approach, but out of the all available techniques, we can always shortlist 2 or 3 formulation techniques to focus on based on the compound properties. It's not we cannot directly choose one technique directly and achieve success in the in vivo studies, but I think we can shortlist which can save the compound and timelines and effort significantly.
Very good. Excellent. Thank you very much for that answer and for all of the answers today. However, we have reached the end then of our Q&A portion of today's webinar. Now, if we couldn't follow or attend to your questions, the team at Aragen will follow up with you after the webinar, or if you have further questions, you can direct them to the email address up on your screen. I want to thank everyone for participating in today's webinar. You will be receiving a follow-up email from XTalks with access to the recorded archive for this event. A survey window will be popping up on your screen as you exit, and your participation is appreciated as it will help us to improve our webinars. Now, I'm about to send you a link in the chat box, and with this link, you'll be able to view the recording of this event on this page, and you can also share this link with your colleagues when they register for the recording here as well. So I encourage you to do that. Now, please join me once more in thanking our speaker for their time today. Sreehari, Putchakayala, we hope that you all found the webinar informative. Have a great day, everyone, and thanks for coming.